Hello friends, today we are going to study about composition, structure and function of carbohydrates part 3. In the last video, we studied about monosaccharides. Now we'll start with oligosaccharides. So what are oligosaccharides? Oligosaccharides are sugar molecules or sugar compounds consisting of 2 to 10 molecules of sugar. Now in that, the most abundant ones are disaccharide. What are disaccharides? Disaccharides, di means 2. So disaccharides are the sugar compounds consisting of 2 molecules of sugar. This is an example of a disaccharide or how a disaccharide is formed. So this is the structure of maltose. Now you see the maltose is made up of 2 glucose. So glucose is a monosaccharide. One monosaccharide. This is second glucose, second monosaccharide. So these 2 monosaccharides are joined by a bond and this bond is known as glycosidic bond. So with the help of a glycosidic bond, 2 sugar molecules are attached or 2 monosaccharides are attached to form a disaccharide, and in this case this is maltose. So what is happening here? This is what the first carbon. This is what the fourth carbon. So in the first carbon, this is an anomeric carbon. Why anomeric carbon? Because on the basis of OH attached with this carbon, we named it as alpha or beta. This is alpha glucose because the OH present at the first carbon is on the opposite direction from the OH present on the sixth carbon. So this is an anomeric carbon. So when anomeric carbon reacts with this carbon is a containing hydroxyl group, OH group, that is alcohol. So here anomeric carbon reacts with an alcohol with the release of H2O. From where H2O is releasing? This OH and one H from here. So H2O is releasing. What is left is only single O. So this single O is here. Hence this glycosidic bond is also known as O-glycosidic bond. Why? Because it is connected with O, oxygen. This is O-glycosidic bond. One more glycosidic bond which we will study in nucleic acid, that is N-glycosidic bond. Okay, so this we will study in the structure of nucleic acid, that is N-glycosidic bond. Here it is O-glycosidic bond. Why? Because this glycosidic bond is joined with O, oxygen. So this is a O-glycosidic bond. When two monosaccharide reacts, during that process H2O is released and hence the bond is formed. The bond is known as O-glycosidic bond. What kind of bond is this? This is a covalent bond. To form a covalent bond, the reaction is happening between the anomeric carbon of one sugar molecule with the alcohol of other sugar molecule. In this case, this is the fourth alcohol. So, in disaccharide, two monomers joined covalently. O-glycosidic bond, that is hydroxyl group of one sugar, reacts with anomeric carbon of the other sugar. Now this glycosidic bonds are readily hydrolyzed by acid, but it does not hydrolyze by base. So the sugar molecules or the disaccharides will break into corresponding monosaccharides if we boil the disaccharide in a dilute acid. One more thing we learned in the last video about the oxidation of monosaccharides, or we learned that monosaccharides are reducing agent. Why we said or why we know that monosaccharides are reducing agent? How we know the monosaccharides are reducing agent? Because if you remember the first carbon, that is CHO, rest all CHOH, right? So the first carbon or the aldehyde group, it get oxidized and convert into COOH. This we learned already, right? So that means addition of oxygen. Addition of oxygen means from some chemical substance it is taking that oxygen. So that substance is getting reduced. So because of monosaccharide, that particular substance is losing oxygen and hence it is getting reduced. Therefore, monosaccharides are the reducing agent. So to become a reducing agent, the oxidation happens at the first carbon. Now, in the case of disaccharide, one sugar anomeric carbon is involved in a glycosidic bond. This anomeric carbon is the first carbon, that is the aldehyde group carbon. So in this case, because now this first carbon is involved in a glycosidic bond for a disaccharide, now the disaccharide become a non-reducing sugar. Why? Because the end which is responsible to behave as a reducing agent is now involved in a glycosidic bond. So it is no more available to reduce any chemical compound. Hence, a disaccharide is a non-reducing sugars. or a non-reducing agent. But still, the disaccharide, or when we study further, the polysaccharides consist one reducing end. Okay, it can consist one reducing end, because for this glucose, if we see, the fourth carbon is involved in glycosidic bond. The first carbon is free, so this can act as a reducing agent. So for a disaccharide, though the whole disaccharide we call it as a non-reducing agent, but it also has one reducing end. For the first glucose, the reducing end is involved in glycosidic bond formation, because here it is first carbon. But for the second glucose, the reducing end is free. The carbon which is responsible for behaving as a reducing end is free. So a disaccharide also consists of one non-reducing end. This side is a non-reducing side, and this side is a reducing side. So a disaccharide also consists of a reducing end. Now let's dwell deep into the structure of a maltose. We learned that maltose formed by the combination of two glucose monosaccharides. A glycosidic bond is formed between carbon number 1 of one glucose with carbon number 4 of the second glucose. So this is anomeric carbon. This is the hydroxyl or the alcoholic carbon. During this reaction, water is released and hence the glycosidic bond is formed. So HOH and H from here released. What is left is only this O. So this glycosidic bond is known as O-glycosidic bond. For the first glucose, as because the first carbon is involved in glycosidic bond formation, so for this carbon, no reducing end is left. But for the fourth carbon, the carbon number 1, which is anomeric carbon, which takes oxygen and convert into carboxylic acid, is free. So this end can act as a reducing end. Now, how we can name a disaccharide? Maltose. It is formed by the combination of two glucose. So this is the first glucose. Alpha, why alpha? We already learned. Why D? That also we know. Glucose, because it is a glucose molecule. Built on the structure of pyran. So the name is alpha D gluco for glucose pyranosyl. This 1 to 4 means it is saying that the bond is formed, the glycosidic bond is formed between carbon number 1 of first glucose with carbon number 4 of second glucose. And the second glucose is D-glucopyranose again. So maltose can also be named as alpha D-glucopyranosyl 1 to 4 D-glucopyranose. Pyranose because pyran ring, gluco because glucose, D because the OH group attached with the farthest carbon is on the right hand side. Alpha, the OH group attached with the first carbon, is on the opposite direction to the OH group attached with the sixth carbon. And this 1 to 4 is the position of the bond. First carbon of first glucose with fourth carbon of second glucose molecule. Its one end is free, so maltose is a reducing sugar. Where we can get maltose? We can get maltose in many fruits and vegetables, even in cereals, wheat, barley, even in sweet potato, we can get maltose. Now next is lactose. Now you pause the video and try to guess the name of the lactose. Rest all is same. This is the structure of galactose, hence galacto. Pyran ring, so pyranosyl. Beta D 1 to 4. Here also you see the bond is between first carbon of the galactose with the fourth carbon of the glucose. So 1 to 4 beta D glucopyranose. This is the structure of lactose. So lactose is a disaccharide which is formed due to combination of galactose and glucose. The first carbon of galactose with the fourth carbon of glucose. So because the second glucose is left with free first carbon, so it can act as a reducing sugar. So lactose is also a reducing sugar. Now one more term is lactose intolerance. You might have heard some people are lactose intolerant. So what do we mean by lactose intolerance? Lactose, whenever we drink milk, because lactose is found in milk. So whenever we drink milk, so lactose enter our body. In our body, we have one enzyme called beta galactosidase. This beta galactosidase degrade lactose into galactose and glucose. This galactose is broken down again into glucose through enzymatic reactions. This glucose is a source of energy. In some adult, this enzyme beta-galactosidase is not produced, or it produced in a very minute quantity. So because beta-D-galactosidase is not found in some mammals, they cannot degrade lactose into galactose plus glucose. So if the lactose is not degraded into galactose plus glucose, this lactose will directly go into our gut or colon. In the colon, bacteria are found. They will degrade this lactose and form CO2, H2O, and some organic acids. These organic acids cause irritation or it cause pain. This is known as lactose intolerance. So the person who are lactose intolerant means in their body the enzyme beta-galactosidase is not produced in proper quantity that can degrade lactose into its component galactose plus glucose. If the lactose is not degraded, then it will directly pass through our digestive system and enter into the colon. In colon, we have bacteria which degrade lactose into CO2, H2 and organic acid. Because of this organic acid, in the stomach, it causes irritation and pain. And this is only known as lactose intolerance. Next is sucrose. Where we found sucrose? It is a principal component of photosynthesis. In photosynthesis, plant produce glucose. We write C6H12O6 only, but that glucose is then converted into sucrose and it is transported in plants also in the form of sucrose only. So it is the principal component of photosynthesis. Sucrose is formed due to reaction between gluco, that is glucose, plus fructose, that is fructose. Glucose plus fructose. Bond is here first carbon, here second carbon. For glucose it is first carbon. For fructose it is second carbon. Now, glucose is a aldose, fructose is a ketose. In case of aldose, the functional group is CHO and the position where it is present first. In case of ketose, the functional group is C double bond O and the position where it is present second. So in case of sucrose, the carbon number 1 which contains functional group and the carbon number 2 which also contains functional group, they both are involved in bond formation. So this sucrose is now not a reducing agent. Why? It is not a reducing sugar. Why? Because both the functional group are involved in bond formation. For act as a reducing agent, in case of an aldose, this functional group takes oxygen. In case of ketose, this functional group takes oxygen. And hence they work as a reducing agent because they reduce the compound from which they are taking these oxygen. So the functional groups are oxidized. In case of sucrose, both the functional groups are involved in the bond formation. So in case of sucrose, there are no such reducing end present. So sucrose is a non-reducing sugar. Hence, in case of sucrose, when we name the sucrose, alpha D glucopyranosyl, you understood why the name is beta D fructofuranoside. Why furanoside? This is furan ring. Now here if you see the arrow, what the arrow denotes? The bond, right? In case of maltose, 1, 2, 4, like that it was written. That means it is showing the bond that first glucose, carbon number 1 is attached with second glucose, carbon number 4. Here the bond is like this for sucrose. It means both the anomeric carbon is involved in bond formation. The 1 is also an anomeric carbon. In case of fructose, this 2 is also an anomeric compound. So both the anomeric carbons are involved in bond formation. Hence, we write arrow like this in case of sucrose, which is a non-reducing sugar. Next, disaccharide, that is trehalose. Glucopyranosyl, that means 1 glucose. Glucopyranoside, that is the second glucose. Bond is formed here also carbon number 1, here also carbon number 1. Hence the arrow is like this. Now here, because glucose is aldose, the functional group is present at carbon number 1 only. And this carbon number 1 is only responsible to act as a reducing end. So in this case, in trehalose, because both the carbon number 1 are involved in bond formation, therefore trehalose is also a non-reducing sugar. Trehalose is the major constituent of circulating fluid of insect. Trehalose is a major component of circulating fluid of insect. So this is about disaccharide. Now one question from my end for you all. In this question, few properties are written. Depending upon this property, you have to guess which disaccharide it is talking about. Okay, question is clear? Few properties are written. I will read these properties. On the basis of this property, you have to tell me that we are talking about which disaccharide. You have to name that disaccharide. What are the properties written? Complete hydrolysis yields only glucose. Clear? Only D-glucose. So if we hydrolyze the disaccharide, the monomers which we get are two glucose molecules, D-glucose molecules. Second, it is hydrolyzed by, here it is missing, alpha glucosidase, but not by beta glucosidase. That means to break the glycosidic bond, we need alpha glucosidase and not the beta glucosidase. How we get alpha and beta formed? That you know, right? So if we can degrade with alpha glucosidase but not with beta glucosidase, what does it mean? That you have to analyze. Third is it does not reduce CO2 plus to Cu. Clear? So depending upon these three properties, you have to name the disaccharide about which these properties are. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.